So this is a topic that I have studied myself, and I think it's really, really important because it can provide such a quick means of improving the welfare of fishes, and yet enrichment is still not universally adopted. So I'm going to start by telling you what we can consider as enrichment and how it can actually improve welfare. I'm then going to focus on laboratory fishes because in many countries we now use more fishes than rats in experiments, and fish in Europe certainly have become the second most popular experimental model. And of course, with the growing populations, there's become a much heavier reliance on aquaculture. So it's important to understand whether enrichment can be employed in fish farming to improve the health and well-being of fishes. So many of you probably think of enrichment as something, putting things in a tank to make it look quite natural. And this picture here shows you the kind of natural environmental biotope that people have it in their homes when they keep pet fish, and also in public aquariums where you try to mimic the actual environment the fishes come from. And you can see that gives lots of spaces to hide, overhang caves to hide in, and also lots of things to swim through. And so we can think of environmental enrichment as making the environment more heterogeneous, making it very different so that the animal doesn't in effect get stressed or bored. Because, sorry to be anthropomorphic, but I'm sure you can agree that being stuck in a barren room for your whole life would be incredibly boring and would affect your welfare negatively. And I think in that case, it is probably the same as true for animals, and there's many studies that show that. Of course, this is laboratory zebrafish. They're a very popular experimental model, and this is the typical kind of housing they're actually kept. And here there's 2 air stones that have been placed in here to provide what's called a behavioral engineering item or a toy, and that promotes novel behaviors in the fish. So they like to swim in the bubbles. But enrichment is more than just physical resources in the tank. It can also be social contact. So who are the animals with, and are those animals fit for them to be together in a social group, or is it a territorial species and they prefer being on their own? So we can think about physical enrichment as well, and that's usually forced exercise or voluntary exercise. So we can think about altering the flows in the tank so the fish have something to swim against. And there are some barriers to adoption, certainly in the laboratory context, and in many zebrafish facilities that keep hundreds of thousands of fish in thousands of tanks. Basically, if you use enrichment like gravel or plants, we have to keep them clean. And so that would involve a lot of staff time, and quite a lot of people are worried about pathogens perhaps building up in the enrichment. So let's take a look at rodents. It wasn't that long ago, rodents weren't provided with any enrichment when they were held in laboratories. And in this review, you can see enrichment can be thought as a number of issues. Social contact, so are the animals held in a pair or a group? Is that temporary or is it permanent? Sensory stimulation, so what visual signals, what can the animals see, what can they hear, what smells do they get exposed to? Is it a new enclosure, is it large, is it complex, or both? Is the food that we're giving the animals the right kind of food to meet their nutritional requirements? How often do we give it, when do we give it, and how is it distributed in the environment? And also voluntary exercise can be provided in the form of running wheels where the animals can engage with the exercise, or we can force them to exercise. And it's known that exercise improves welfare. And then we can provide novel objects or behavioral engineering items where we try and encourage the animal to perform novel behaviors. So things like colored balls and toys that the animals can play with almost. So this is really trying to prevent boredom in the animals. And this kind of enrichment is now adopted as standard. You would never keep rodents in a barren environment in the laboratory. And the reason for that is when you provide all these enrichments, it leads to good mental and physical well-being, good welfare. However, when you keep them in barren environments, this can lead to psychiatric disorders and, of course, stress. So really, the studies on rodents show us that enrichment is really good for the animals, and that, you know, we don't want to be doing experiments on animals which are stressed or have, you know, nervous system problems or disorders. So I talked about zebrafish. They are probably account for 50% of the animals that we use, or I should say the fish that we use in experiments. So for example, here in Sweden, around 60,000 fish are used per year, and 36,000 of those are zebrafish. In the UK, they use something like 500,000 fish per year, and 50% of those are zebrafish. It's a very important model. And here's the natural environment. You can see there's lots of crevices, plant life, and this is a typical environment you would find zebrafish. They're Asian, and you can go out and collect them in the wild. However, we keep them in an environment that is very, very different. And this is a typical barren standard tank. And you can see this is a zebrafish facility with lots and lots of tanks lined up together. And many people believe that because they're a gregarious, sociable species, we only need to keep them in groups and that's enough. However, I wouldn't agree with that. So why are zebrafish used so much? Well, they have all of these wonderful advantages, and they are being adopted across biomedical studies. So the study of clinical disease in humans, zebrafish are now being used as a model. And one of the main things is that they are more economical, they cost less than rodents. And so many researchers are now turning to the zebrafish model, and that's becoming an issue in terms of more and more zebrafish are being held in laboratories. So really we need to know whether they require enrichment. This is a survey done by Katie Lidster and colleagues, and this is the number of survey responses from the different countries. So you can see that across the world, people are using zebrafish, and they're using them in such a wide variety of studies, developmental biology, neurosciences, zoology. And so they're used across a range of experimental contexts. So I would say their welfare is particularly important. So zebrafish facilities can house hundreds of thousands of different strains of zebrafish, and to be able to go around all these tanks and check the welfare of the fish twice a day, and also then implement enrichment, can be challenging. And so in the zebrafish facilities, enrichment is not always adopted. So what we need to do is actually find out whether zebrafish need enrichment and what would they choose. And we've done that by running preference tests, by giving the fish a choice between resources. And then we need to ask the question, is this good for the fish? Do they actually benefit from that enrichment? So here's just an experiment I ran in my lab where we put zebrafish in the start box. This is a really big tank. Zebrafish are held in barren tanks with bright lights. So we had a side which was barren and brightly lit. On the other side, we had gravel, no bright light, we had a plant. And then because they're sociable, we had a shoal of zebrafish behind a transparent wall. And what you can see here is a zebrafish just right here in the enriched side. And this is just a video showing you fish swimming around. Sorry, this video is getting a bit old, I think, and it's quite difficult to see the fish, but I'll just follow it with my mouse. So you can see the fish, the doors are open, and then the fish makes an entrance, goes straight over to the shoal. Now these fish were held in and reared in barren standard tanks for 7 months, and yet they always chose enrichment. So that would give us an insight into the zebrafish mind, if you like, and show that they actually prefer enrichment. So then I had a great guy come and work in the lab, Paul Schroeder, who did his Doctor of Veterinary Medicine with me, and we decided to ask the fish what they want. So we just used a standard housing tank, and we put in a perspex divider with a little doorway, and then we could put resources on either side. So we have the barren conditions here, and then there's gravel on the other side. And so we decided to build up a preference order by pitting the resources against one another. We looked at the impact of sex, whether male and females prefer different things. We looked at dominance relationship in pairs. So zebrafish are very aggressive to each other and small group sizes. And then we looked at the group preference. So obviously stockfish are held in groups, and fish are also held in breeding pairs. So we had the male-female pair, and then we had 4 male, 4 females in the groups. We gave them sand or gravel, or actually an image of both. And we did this because of the hygiene issue. We then used a submerged plant and a floating plant. And then we used an air stone as a kind of toy. By pitting the resources against each other, we could work out a preference order. And this is just one of the experiments, as Paul filming the fish. And you can see most of them are in the enriched side. And so we came up with the preference order. So here we have the group males, the group females, and then the dominant member of a pair and the subordinate member of the pair. And the groups, we can see that the males prefer gravel, even the gravel image over sand, and then the floating plant was next. Females seem to prefer sand, and then the floating plant, but also the gravel image. In terms of the pairs, the dominant fish was the largest fish, which is the female, so that kind of confounded things a little bit. And the dominant basically chased the subordinate out of the preferred area. So again, you can see the female like sand and a floating plant. And then they actually avoided the air stone, so the poor subordinate was forced into close contact with the air stone. So we can see there is a difference between the animals in groups and between the sexes. However, the gravel image and the plant comes up quite a lot. So this gravel image, I use it a lot in my lab, and I've been in other labs who use it. And what you can see here is the gravel picture, the amount of time, the percentage spent in the area with a gravel picture, and then the darker grays in the barren side. So both males and females prefer the gravel picture to a barren side. It's not quite as clear cut. The males seem to prefer the sand picture, but not the females. So going forward, I still use images of gravel in the lab now to help improve fish welfare. And I approached a company who will remain nameless and asked them, can you produce tanks with a gravel bottom? And at the time they said, no, we're not interested. Of course, lo and behold, they're now producing gravel bottoms. So these can be inserted underneath the tank. And this is their latest tank, which has a gravel bottom. And they cite my paper on the website. So I don't get any money, but I am pleased that the research has an impact. Other studies have looked at preference for flow and enrichment. So here, when you have flow, you force the animals to exercise. So in here, the fish were placed in a neutral central area. Then they had flow, or they had nothing, or they had enrichment, or they had flow and enrichment. And then this is the preference index. So at 0, you have no preference. If it's a positive value, you have a preference, and a negative value means you avoid. And you can see that the enriched plus the flow side was most preferred, followed by the enriched only, followed by swimming, although there's no significant difference between that and the plain barren side. So there is a preference for flow. You can also look at other biological indicators. So for example, we can set up zebrafish breeding pairs, and then you can collect the eggs really easy. It's external fertilization the next morning, and you can test things like grass or leaves. And here we have the number of eggs in the control, barren conditions, grass and leaves. And you can see there's significantly more eggs when grass was present. Then we can ask, does enrichment help survivorship? And so at very young ages, there is quite a high mortality rate in young fish. Carol Lee and colleagues at Exeter looked at this. So Carol reared the fish from 5 to 30 days post-fertilization and looked at survivorship at 30 days. And again, when they were reared in rich conditions, we get over 80% survival, whereas in barren conditions, it's around 50%. We can also ask the question, does enrichment reduce anxiety? And this is a classic anxiety test. You basically put the fish in a novel tank, and then you record the amount of time spent on the top of the tank, because fish, when they're anxious, will sink to the bottom and won't come up. So if they go more in the top, they're less anxious. And again, we see this effect of enrichment. They spend much more time in the top half of the tank than fish which are reared in a barren environment. We can look even further into the sort of the brain mechanisms. In this particular study, the authors looked at keeping fish in an enriched environment like this, or in a barren environment, and then found how many nuclei were in a specific brain region, how many new cells were being formed. And here we find that in an enriched environment, we get many more new cells than in the barren environment. So brain development is better under enrichment. And so there's lots and lots of evidence that enrichment really does improve the welfare of zebrafish in a range of contexts. And so why is it not widely adopted? And again, it's this idea of a lack of hygiene and staff time at implementing it. However, I'm hoping that with more research, we can convince the zebrafish community to adopt this. And if you want to read further about this particular subject, I wrote a review on the ethical considerations of fish research with Cass Lohman, Ian Boyukos, and Ed Brooks. Let's move on to aquaculture. Aquaculture is starting to be adopted in fish farming, but it's very slow. What you have here is inside a salmon cage, and then here we have these kelp rings, which are introduced to mimic kelp. And the reason they're there is for these little fish here, these lumpfish. They're called cleaner fish as well. And lumpfish are used to remove lice from the salmon, and it's thought to be a natural biological control mechanism. Of course, before the enrichment was introduced, these poor lumpfish were perishing in very high numbers. And this is because they have a sucker on the bottom of the body, and they like to cling on to things. But by introducing these kelp rings, this has improved the welfare of the lumpfish. And just to show you, these are not salmon, but this is a natural kelp bed. So it does quite mimic that. We can also look at trout farming, and rainbow trout, for example, are the #1 farm species in Sweden. This is the normal environment. You know, we've got a pebble bed and places to hide, and this is a typical trout river. You've got different areas of flow, lots of overhangs, and then we look at a trout raceway, which is very barren, and this is a recirculating aquaculture system, and you can see a very high stocking density, but no enrichment is employed. So we looked at this question in my lab with a postdoc called Kieran Pounder, and we looked at rates of recovery when the fish were held in either enriched conditions. So whenever I do experiments on behavioral ecology, applied animal behavior, ethology, I would always have gravel, plants, a tube for the fish to hide in, and overhead cover. But many other people do their experiments in barren conditions. So we compared the responses from trout held in either barren or enriched conditions. So what you can see here is the recovery rate of the opercular beat rate. So when fish become stressed, they breathe really heavily by pumping the gills. And the higher the rate, the faster the fish recover. So when we imposed stress, we saw that the fish recovered much more quickly in the enriched environment than the barren environment. And the same when we anesthetized and handled the fish, it recovered much more quickly if it had been in an enriched environment. So let's move on to another example, and this is an example from sea bream, and it was done by Pablo Chavala Lopez and colleagues, and they did a very clever experiment where they used plant fiber ropes. So they effectively tied the ropes to the bottom of the cage, and then they used buoys to keep it upright in a vertical position. And this is just a look inside these cages. And so they introduced this very simple enrichment. They looked at petrel fin damage at day 0 and then 35 days afterwards, T35, in enriched cages and non-enriched cages. And what you can see here is actually there is a reduction in petrel fin damage with the use of enrichment, whereas the petrel fin damage stays the same when there's no enrichment. Then they looked at the impact on the caudal fin, and we can see again here, there is in both cases an increase in caudal fin damage, but not to the same extent in the enriched sea cages. They then looked at behavior, so they looked at aggressiveness over weeks 1 to week 5. And if I just draw your attention to this part here, this is the total aggression over the entire trial. Under enrichment, in this grey bar, we have less aggression than when no enrichment is present. So that is a positive outcome. And then this is the number of fish interacting with the nets. And again, with enrichment, we don't see so many fish actually interacting with the net than the non-enriched fish. And this is where they can get abrasion from the actual net. Pablo also looked at the use of the space of the sea cage. So this outer ring represents the edge of the sea cage, and then this inner dashed line represents the middle. And what you can see here in the colors is the percentage of fish using these areas. So you can see when it's non-enriched, we have this white area, so no fish are going into the middle, whereas there's much more middle use when the enrichment is present. And this could be similar to what's called thigmotaxis, wall hugging in mammals, where you basically have anxious animals hugging the walls. When they're less anxious, they'll explore the middle of the cage more often. And then they looked at increased survival, and under enrichment, there was around 75%, and with no enrichment, there was around 65%. So basically 10% more fish survived with enrichment, but this was non-significant after statistical testing. And perhaps if the experiment lasted longer, we might see a change. Other studies have looked at Atlantic salmon and brown trout from different populations. And this is freshwater salmon stage, so these are salmon par. And then all they did was place some pebbles inside the tanks to enrich them. And then there was a natural infection outbreak. And what we have is the brown trout population. So we looked at 5 different populations and then 5 different populations of Atlantic salmon. And what you can see here is in the white bar with the dots, that's the enriched tanks. And then the gray bars are the standard barren tanks. So mortality of the trout was much higher in the standard tanks and almost negligible in the enriched tanks. And then when we look at salmon, again, we've got very high mortality in the standard conditions and much lower across the board for the enriched conditions. So the enrichment actually improved these animals' resilience to an infection, and they actually recovered. Then they took 2 populations of brown trout and then exposed them to a bacterial infection and looked at survival rate. If the animals had been reared in enriched conditions and kept in enriched conditions during the challenge, or if they were in the barren standard conditions and then placed in enriched, or reared in standard, kept in standard, or reared in enriched but then placed in standard. And what you can see here from this population, the dark line, enriched fish kept in enriched conditions have much better survival than those that were reared in standard conditions then placed in enriched, and certainly more than those held in standard conditions. We see a similar pattern for this population as well. So environmental enrichment appears to have a very significant positive effect on the welfare of aquaculture species, and I've given you some examples to illustrate that. What kind of things should we be concentrating on? Well, we need to think quite clearly about the physical enrichment of any aquaculture tank or sea cage. We need to see, are the structures there providing shelter or a heterogeneous environment? What substrate should be used? If the tanks are on land, can we use sand, gravel, or pebbles? And that will differ between species. So we get flatfish which live on the bottom of the tank. They will require a different substrate to fish which are pelagic and swim in mid-water. Then how do we occupy the fishes? Do we alter flows? Can we alter the water movement? And in that case, can we get the fish to swim? And forced swimming doesn't sound so nice, but actually the benefits of exercise are quite clear in fish, that they grow better and convert feed better. Then we need to think about the sensory environment. What about visual disturbance or visual cues that we give them? And in particular, we need to think about the lighting. what kind of daily rhythm are we giving them? What wavelength of light are we giving them? Are we giving them shelter if the light is bright? So that's a consideration. And then there's lots of sounds in aquaculture facilities and sea cages. Obviously, pumps are going off. And can we reduce any stress with sort of unpredictable background sounds? And there has been some studies showing that actually playing music constantly can improve the welfare of fishes. Then we can think about chemicals. You know, fishes live in an underwater world, and the chemical smells and things are very important to them. So we need to think about whether pheromones from other fish are accumulating. So fish do send out signals in urine and other excretions, which can signal dominance. Does that build up, and is it bad for the fish? Or can we use positive pheromones to improve welfare? Then tactile touch stimuli. Cleaner fish do eat off these lice from salmon. Do they also provide a tactile stimulus that's positive? Can we use different hydrodynamic flows to basically provide a touch stimulation or stimulate some of their sensory systems? Then in the social context, we have to give the right social environment for fishes. We have to think, are they territorial or are they sociable, and how the presence of others affects them. We're really looking at having low aggression in these tanks and low stress. We can also think about provision of appropriate reproductive partners where that's practically possible. Then we need to think about space. How many fish are in the tank and what is the stocking density? Does the fish have the amount of space that it needs so it can swim and turn and be unrestrained? And finally, an appropriate diet is key for animal welfare. Are we giving the right nutrition to these animals? What is the content of the food? Is it tasty? Is it floating or does it sink? Should we give a variety of this? And hatcheries, we often use live foods like invertebrates like Artemia or rotifers to actually feed to these fishes. Is live foods acceptable? It's a rather ethical dilemma. And then what is the regime? When do we feed them? How often do we feed them? And can we use self-feeders? Can the animals actually feed themselves and get food when they want it? So these are all the things we need to keep in mind to enrich the environment of fishes. And thankfully, Pablo and his colleagues have come up with this scheme for moving forward implementation of enrichment in aquaculture. And I think we can also apply this to the laboratory situation. One of the things we have to be very careful with is which fish species, because each fish species, and there's something like 35,000 of them, have very different preferences and different needs. And also developmental stage affects their environmental needs. They also need different specific things in their environment to meet their behavioral needs, and they may have different preferences or motivations. So we need to take all of that into account, but then we need to be in partnership with the fish farm and look at stocking densities. What kind of farm is it? Is it a sea cage? Is it a RAS system? What kind of facilities do we have? Then we come up with a design for enrichment and we develop it. We want it to be easy to implement. We want it to be hygienic, and we test it out. And often that's done in the lab or in one farm. If we get undesired results, which often happens in science, we go back to the drawing board. However, if we get good results, we can then go large scale and using different farms. We can then look at cost benefit assessment of implementing it wide scale. And then hopefully this would lead to the industry adopting this enrichment, and we can long-term monitor the impact on fish welfare. Now, there are many benefits for the fish. There's good health and welfare. They're allowed to perform natural behaviors. They then are more resilient and can cope with disease and stress better. And so their welfare is improved. But also in partnership with a fish farm, there is lots of reasons why you'd want your animals in good welfare. They grow better, they're resistant to stress and to disease. They then have better flesh quality. You're not losing them if you do get a disease challenge. And we can improve the rearing standards. And of course, it's the public that demand good welfare. And so fish farms can show that they take this very seriously, and that can really improve their public perception. And of course, Pablo and his colleagues, they're based at the Fish Ethology and Welfare Group. They have produced a fantastic review. So I've only touched on a few examples, but it's really a wonderful read if you're interested in this topic. They cover many different enrichments and many different species. So just to conclude, the use of fishes is growing across the globe, either in science or in aquaculture. So this is a really great moment to start to really try and improve the welfare practically by employing enrichment. I hope I've shown you that environmental enrichment can have a very positive effect on welfare, and we can actually make a substantial increase in the animal's resilience and biological functioning. Now, as I said, there are many other species that are used in the laboratory and in farms, and I really do think it needs to be on a species by species basis, so a lot more research is needed. And we also need to consider differences in developmental stage. So for example, salmon start off in the freshwater and then they migrate to sea and then come back. You know, their needs are going to be quite different. We also have to think about sex differences, and then of course, which kind of aquaculture system or laboratory facility is available. And then I'm hoping that we can develop new environmental enrichment tools to implement them across laboratories and in aquaculture. And I think the implementation barriers is, and I hear this a lot, there's not enough research, not enough data, it's going to cost a lot of money, and it's going to be unhygienic and threaten biosecurity. But I hope I've shown that that's not necessarily the case. And so to move this forward, I think research funding bodies really need to make environmental enrichment a priority. It's very applied research, but it's no less important. It can really improve the lives of a large number of animals. So just to thank my funding bodies, my collaborators, and the wonderful people I had working in my lab. And just to thank you very much for your attention, and I thank the Aquatic Life Institute for inviting me here to talk about this topic. Thank you.
Thank you so much, Dr. Sneddon. I don't know about the audience, but I can definitely listen to enrichment interventions and research all day. Yeah, that was great. Thank you so much. We do have a few minutes, so I'm going to pose some questions that we have. The first one, since environmental enrichment is primarily based on sense perception, are there aspects of fish perception that are more responsive to enrichment? And what sort of activities or interventions are best?
Yeah, I think fish, you know, obviously live in a watery world. So in terms of the senses, their senses are attuned to that underwater world. And of course, we always think as vertebrates living in terrestrial environments, you know, we can kind of understand what terrestrial farm animals would like in their environment. So it's something we need to think about. Fishes have an extrasensory system, the lateral line system, where they can detect the direction of movement of water, and that's why you know, altering flow might be quite stimulating for them, you know, to sort of stimulate those systems. They also live in a very chemical world. They use chemical communication a lot, and so it's important that we prevent the buildup of any sort of negative substances, and in particular, you know, pheromones that might signal dominant status. That'd be stressful to subordinate fish. People have looked at the buildup of cortisol, because fish are excreting cortisol over the gills. I think the species that have been tested, it hasn't had a great effect, but it is possible that it might do. So yeah, I think we need to think a bit more like a fish, and try and use enrichment that really benefit them. So we need to get a bit more fishy.
I love that. I'm gonna take that one for later. We need to get a bit more fishy. Great, thank you. And the next question, what kind of enrichment strategies merit more attention? For example, structural, sensorial, occupational, social, or dietary enrichment by the industry? And do you see it being mandatory in the future?
Yeah, I think all of those are equally important. I think prioritizing, I think we need to think about what is easy to implement and what are the quick wins. And so, for example, the ropes floating in sea cages, they were a fantastic quick win, really easy to do, really easy to implement, cost effective. So physical enrichment can provide sheltering, it can stimulate normal behaviors. And so I think, having some control over their environment is very important for fish. And if they have places to hide and places to take shelter from either dominance or from, staff passing by, I think that's really important. So physical enrichment, I think, is one that is very quick and easy to implement without too much effort. But of course, then we need to work out what each species really needs. I would hope that environmental enrichment does become the standard because, you would never keep any of these other animals, really. Chickens, pigs, cows don't get kept in a barren room with nothing there. But yet we seem to not apply the same kind of considerations to fishes. But I'm hoping things are changing, and certainly with this conference, we're seeing people talking about this very important subject. So I would like to see it become standard.
Yeah, definitely. I would as well. I guess along those same lines, have you spoken with producers, and are they in favor of using enrichment, or have you experienced some level of resistance?
Yeah, in terms of aquaculture, I've spoken to quite a few of my colleagues about this and with farms, and I think if you can show benefits, they are very amenable to implementing enrichment. But it's providing sort of the research data and the evidence can be quite challenging. It's very applied work, so it's very difficult to get funding for this kind of thing. But I know, talking to colleagues in Switzerland, that many farms want to go organic for trout farming, but then they have to use gravel. And some of them have reported that if they have slow flow, that then uneaten food and feces sort of collects, and that can lead to pathogen outbreak. And so then they're a bit resistant to employing enrichment for those reasons. But yeah, I think on the whole, people are receptive if they think it's going to benefit their business. But it really is all down to providing them with sound scientific evidence and really showing them that this is an easy thing to do and that it's practically possible and is not going to cost a huge deal of money to actually do it.
Great. Thank you so much, Dr. Sneddon. Unfortunately, we're out of time for this session. Like I said, I could sit here all day, but I just wanted to thank you so much. And thank you to our amazing audience members for joining as well. Please keep out an eye for post-conference communications, because we have different call to actions and related to ways you can continue helping aquatic animals. And we also have a very exciting announcement that Dr. Sneddon will be one of the guest editors for a special issue of the MDPI journal Fishes. So we will be sending out more information for that as well after this conference. Thank you again, Dr. Sneddon, and thank you to our audience. A quick reminder that our next session is starting now in about 9 minutes, and we will hear from legendary Dr. Sylvia Earle. And we hope that you all can stay logged on with us. Thank you so much.
Thank you. Bye.